Professor Nana. I'm Andrew, and I'm a research assistant at the Microbiota Eye Center in Hong Kong. And it is our great pleasure today to have you sharing your research experience with us. And so, with no further ado, the first question we have for you is, why are you so interested in microbiology, and what makes microbiology so appealing and fascinating to you?
Yeah, thank you for the question. In college, we took classes in many fields of biology. I found microbiology so fascinating because tiny organisms can have such a big impact on our world. When I started my PhD training, I further learned that microbes could further play an essential role in human health and disease. I'm so interested in learning more about how microbes affect their host. We need to know how bacteria engraft in human body, how they interact with each other, and how they interact with their host. So we can use this knowledge to improve human health care and nutrition. All of this developed my interest in this field of microbiology.
I see. So just like you mentioned, and you're particularly interested in the interaction between humans and microorganism. And so for which area, which field, which research field attract you the most?
Yes, the specific fields I'm interested in are mostly the host and environmental factors' significant effect on the structural and also functional contributions of the microbiota and human health and disease. I'm interested in investigating the associated and also causal relationship between functional gut microbiome and also human disease, for example, obesity, diabetes, national food, IBD, and so on. And I'm also interested in applying opportunistic gut pathobiology for predictive biomarkers and targets for therapeutic interventions, and also investigating the effects of diet, probiotics, and prebiotics relevant to the future of precision management of human health and disease through modulation of the gut microbiota. Yeah, I think I'm interested in this kind of field.
I see. So could you share with us a huge project or research that you've done or accomplished, and what steps did you take to do the project, and how did it turn out?
Yeah, the largest project actually I completed happened in my PhD training. That project tried to address the chronic inflammation induced by the dysbiosis gut microbiome, contribute to the development of obesity-related metabolic disorders. This project included 123 central obese cohorts. In this project, I served as primary investigator. The pressure was on because we had to pursue the subjects with obesity to accept the strict dietary intervention and also maintain the intervention for the whole 23 weeks, which is a very long period. Another challenge was that we want to apply germ-free mice to investigate the causal contribution of some bacteria isolated from this obese human gut. And it was very difficult to maintain the sterile status of the germ-free mice at that moment. So for the team, as well as for me personally, these are big deals, and the project needed teamwork and also a professional and careful attitude toward technique. So with an experienced team, planning documents first, setup, goals, and milestones were determined at each project stage. So we delegated the task based on the knowledge and also experience levels of each team member. Also, we carefully monitor and discuss together the milestone progress. So with the teamwork, we finally finished the project. We successfully showed that one endotoxin producing bacterium isolated from a morbidly obese human gut can induce obesity and also national food in germ-free mice. We also published a series of papers in related journals.
All right. So with all the research you've done, so what is the most exciting research project you've worked on, and why did you enjoy it so much?
Yeah, that is a very interesting question. I have worked on a variety of projects in my career in microbiology. I found that each of the projects are very challenging to me, and I certainly learned many things from each one. So instead of considering projects most exciting to me, I consider each one as a learning experience for me. For example, in the first project, I introduced just earlier, I studied the relationship between microbiota and obesity. In that project, I learned how to work with human subjects, and I learned how to use the dietary intervention to modulate the gut microbiome. And I also learned how to apply the germ-free mice to investigate the causal contribution of the microbes to human disease. And for another example, in another project with a team collaborators, I lead the research that define the association between the oral and fecal microbiome biomarkers and cardiovascular risk in a cohort with 655 participants of African origins. In this project, I actually learned how to study the microbiome through bioinformatics analysis tools. So I mean that each project I worked on was very exciting to me, and it helped me to increase my knowledge in this field and also helped me to move forward in this field. So I feel that all the projects are exciting to me.
I see. So can you tell me about a time where you had to like gather different data from multiple sources, and how did you manage to do that?
Yes, to study microbiome, actually, you have to collect multiple metadata for microbiome. For example, the 16S RNA gene sequencing, the shotgun metagenomics, and also the assembled genomes, the metabolomics, and also networks analysis of microbial communities to investigate the structure and also the function of the whole microbiome. We are trying to answer who are there in our body and what they are doing. So for most of my current projects, we have to gather all of these multi-omics data by working with different centers and also using different types of sequencing platforms. I have the ability to study the microbiome through the bioinformatics analysis tool, and we also cooperate with many experts and also professionals in related fields to accomplish sophisticated bioinformatics analysis. So the whole goal to collect together all of this data is to explore the molecular crosstalk between the host and the microbiome.
Yeah, I see. So like Magic is one of the center you've been working with. Yeah. And I believe like very often as a scientist, we have to utilize lots of like information and sometimes like state of the art, a completely new piece of technology on your own research. And like how was it? Like was it difficult to like merge the technology together to your research? Is it difficult?
Yeah, actually, I found it very exciting to me to learn new things and also new tools in my projects. I keep on changing projects and keep on learning. The most impressed one is the network analysis for me. So in one of the projects I lead in the past, we wanted to further probe how the microbes might interact with each other and also the microbial community. So in that project, the microbial community was quantified and compared using co-occurrence network analysis. We are trying to predict the interactions between the microbial community members and also to explore how this network structure might impact the community stability and resilience. So in this model, to evaluate the predicted resilience of the network, we applied attack tolerance on these bacterial co-occurrence networks. And in this project, we found that this technology applied in this project demonstrated that the host specimen microbiome network exhibits high robustness on targeted attacks. However, the exogenous network structure degraded rapidly and appeared more vulnerable. So this is a piece of new techniques I applied in my project, and this is the most impressed one.
And like also, I think as a scientist, like we have to expect failures and obstacles during the whole process. And because not everything stick to the original plan and go as we thought it would be. And how did you like share a undesirable results with the public or other professionals?
Yes, actually, that is always happen with all the projects. We cannot make sure that all the projects succeed all the time. As you said, we fail sometimes. So in my research, most the failure experiment happened with germ-free mice. The germ-free mice are one of the most popular animal models we use to study the function of the microbe in human disease. And the most important skill required in this model is to protect the germ-free mice from contamination. In the past, it is a very strict technique needed. Because of this, we weren't able to accomplish the animal studies or get the results within the expected time sometimes. I had to tell the frustrating news to the researchers or collaborators in the team when the contamination happened. And after explaining why we need to take a very long time to confirm the sterile status of the mice, and we also need to repeat animal experiment to make sure that we got the precise and the right results and conclusion. The other researchers in the team agreed that the sterile status of animals was the most important factor in the developing of any mice model. And they become, I think, understood. I think that will be very helpful.
Let's see.
Mhm.
So I think that's all the questions I have for you today. And to be honest, I think it is a great meeting, and it really showed me how a research goal and progress into a like a to what we wanted. So I think that's it for today, and I look forward to meeting you in Hong Kong.
Look forward to
And
meeting you too. Thank you so much for your time.
thank you.